So now we come to one of the early career researcher programs, which is we gave them the opportunity to submit their abstracts and present their work in front of all of us. And we were so happy to receive many, many really good quality abstracts. It was a tough decision to choose just a few to be represented on the stage. However, we've chosen 8. And today we are going to have 4 of them talking about their work here. So we will begin with Devashree, followed by Ami. So Devashree is a PhD student in the laboratory of Dr. Prajakta Jain. And she works at Institute of Chemical Technology in Mumbai. Today she's going to talk about retinal cells and how they have used these as a microfluidic device, essentially retina on chip.
Thank you so much for the introduction. My name is Devashree Jahagirdar and I'm a PhD student working in Institute of Chemical Technology under the guidance of Dr. Prajakta Jain. So today I will be presenting on microfluidic platform for development of bioartificial retina. So all the conventional methods we already are in discussion with since morning, that the conventional methods has several limitations like major animal sacrifices, ethical issues, economical unfeasibility. Then they do not reproduce as it has to be done physiologically. And sometimes they are compromised with the high throughput screening techniques, and they do not reproduce well. So the prevalence of retinal diseases, only 40 to 41% of the population has normal eyesight or retinal health. Almost 60% they suffer from gradable or non-gradable retinal diseases. And as we are all familiar with the drug failure rate, the maximum drugs which get into clinical trials or preclinical trials and further move on to phase 1, phase 2, phase 3, there is a maximum probability of drug getting failed at even phase 3 stage. So we have very less amount of drugs which are coming actually into the market, and we can actually use it for ourselves. So moving on, as Indumati ma'am has already brushed up about the retinal cellular layers. These are the cellular layers which are there in the retina. So the most crucial and most sensitive layers are the photoreceptors, the RPEs, that is the retinal pigmented epithelial, and the endothelial cells. So in this study, I have been focusing on using retinal precursor cells instead of photoreceptors to understand how exactly biologically they are responding, and retinal pigmented epithelial cells. So why use microfluidic technique? We are already familiar with the conference being conducted, that it provides a high throughput screening, it provides perfusion, and it has high translatable value. And most importantly, it allows us to have a operational control. We can modulate it as per we want. So here we have developed, designed and fabricated a microfluidic model in collaboration with Abhijit Majumdar sir from IIT. And the model is a 2-chambered device which is connected together by a tubing, and we have each chamber for cultivation of each individual cell types. So here we are using retinal precursor cells and retinal pigmented epithelial cells. So this model has been undergone with the computational studies as well to check the pressure, if the pressure is sustainable by the cells, and also to check the shear force if the cells have been under any shear stress. We have performed media optimization because since we were using 2 different cell types, we have to have a single media for culturing of them. And we have characterized them with several cellular markers. Like for epithelial cells, we have RPE65. For precursor cells, we have characterized it with recoverin and PAC6. So moving on, we studied the co-culture for glucose consumption because it has been already reported that most of the retinal cells consume heavy amount of glucose. So we have studied or quantified the glucose consumption. We have also studied the permeability coefficient for single cultures as well as the co-cultures, wherein we found that the permeability was lowering in the co-culture system. We have also quantified for inflammatory cytokines for single cultures as well as co-cultures, and we have tried to induce inflammation in all the 3 sets with using lipopolysaccharide. And as we can see that in the normal conditions, the inflammatory cytokines are quite less. Further, we have tried to study the differentiation of the precursor cells into the photoreceptor cells. So under the influence of epithelial cells, since the RPEs are they act as a feeder layer for photoreceptors. So we have studied for rhodopsin expression in precursor cells, and it has been seen that they express rhodopsin quite nicely. Then we have exposed these the differentiated photoreceptors to violet blue light, and we have seen the effect of the light exposure onto the cells, and it has been seen that blue violet light, it has a tendency to cause intracellular stress, and therefore the dead cells were increasing in the light exposed sample. Also, we have studied for the inflammatory cytokine after exposure of light, wherein we can see the TNF alpha value was increased. And we have also quantified the intensity of rhodopsin in the photoreceptor cells, and as reported, it was very well validated that the rhodopsin expression after the light exposure was lowered down. So with these assays, we were able to at least perform a validation of the device which we have already developed, that okay, this device can accommodate co-cultures, and it can also give us multiple screening techniques at a time. So we can real-time image the cells while they are growing, while they are undergoing stress, and while they are dying as well. And during the whole process, we can aspirate the outlet medium and check for the inflammatory cytokines as well. So moving on to the second and very brief explanation of another model, which was the in vitro triple culture model of retinoblastoma. So I have developed this model on a transwell. It is a triple culture model comprising of retinoblastoma cells, retinal epithelial cells, and the endothelial cells at the bottom. So RPE cells are plated as an inverted way on the cell inserts. So this is how the culture looks at after imaging. So the yellow ones are the retinoblastoma cells, these are the RPEs, and the red ones are the endothelial cells. So we have devised a new drug concentration to control the retinoblastoma cells without affecting the RPE as well as the endothelial cell growth. So here we can see that Y7, that is the retinoblastoma cells, were decreasing. So what is BM? BM is bevacizumab, and in combination I have used carboplatin. So with the increase of carboplatin and bevacizumab, we can see that the IC50 was achieved at almost around 12.5 microgram per ml of carboplatin. And at this concentration, only the retinoblastoma cells were observed to be possessing the death phase. And it was not affecting the epithelial and the endothelial culture, as we can see here. So these 4 images are of RP and the endothelial cells. Moving ahead, we have studied for tight junctions also. So these are the tight barrier properties. We have also studied for vimentin expression. Since it is a tumor model, we have studied for the vimentin expression as an EMT signal, and we see that at the combination of bevacizumab and carboplatin, the EMT signal has been lowered down. We have quantified inflammatory cytokine TNF alpha here as well, and we see that after the combination treatment, the amount of inflammation was reduced. So this triple culture model also supports the, also selectively kills the retinoblastoma cells and not affect the rest of the co-culture, as well as serves the very good barrier properties. And the third and a very short topic which I was covering in my PhD was to mimic the disease, that is AMD, by preparing a disease induction cocktail which will serve as reactive oxygen species generator, lipid accumulator, since usually the so this is the normal physiology, and after the, just a minute. Yeah. After the stress inducers, the photoreceptors tend to shred off, as well as there is Drusen deposition that impacts the RPE cells and disrupts the whole lining. And then there is the wet AMD, in which the blood vessels erupt and there is internal bleeding in the eyes. So this is just a short study which I am still conducting, that this is the co-culture for epithelial and endothelial cells. So after the disease induction cocktail, what happens is the barrier integrity property has been compromised severely. Along with it, this is the fresh study. So lipid deposition in the intracellular cavity as well as in between the RPE and the endothelial cells have also seen to be increased. So the cocktail which I have devised here is working quite well. So with this I would like to conclude. I would like to thank DST, DBT and ICMR for the funding. And the current ongoing work in our lab is skin on chip, which you will see in Ms. Deepa Chaturvedi's poster, that is poster number 11. So yeah, it has been used for dermal epidermal co-culture. Thank you so much.
Thank you, Devashree, for the eye-opening talk. Would anyone have questions for her?
So in your co-culture, the chip co-culture model system, so what is the rationale for this? Though you say it is a co-culture, you are actually putting the cells in two different compartments.
Yes.
Only the fluidics are connecting them.
Yes.
But actually these cells do directly interact. So
Yes.
if you have to relook at your system, so do you, how do you plan to?
So the initial rationale behind the device design was to connect it through microchannels so that we can see the neurite interaction as well during the co-culture, because precursor cells tend to extend their neurites towards the RPE cells to interact or create a biochemical gradient.
So that you will not be able to look at in your two-chamber system,
Yeah,
right?
it is not, like these studies do not have that particular parameter in the device, but we are currently working together with Abhijit sir for development of these microchannels in it.
So another question about your AMD model. So how are you inducing the AMD condition?
Sorry?
How are you inducing the AMD in?
So, I have focused on different parameters individually, like the lipid one, the ROS one, whichever more long, like it will really keep releasing ROS species for a longer time, like keeping the not actually killing all the cells at once, but just allowing only some cells to. So I'm optimizing the concentration. I have optimized the concentration to such an extent that it will only erode off some cells and not all of them. So there are different compositions in which I'm giving or inducing the stress in them.
So in continuation to ma'am's co-culture question, so when you say co-culture, you add all the cells at the same time, and they grow together, or you add one after the other in that model?
Okay, so in these the cells in chamber 1 are added primarily. Yes, so I coat the device first using protein mixture, and then I add the cells for the adherence of it. Then I add first the epithelial cells and then the precursor cells. I let the perfusion system for keep it on for some time, and then I add the precursor cells to it.
Just a small query. Why did you choose 405 nanometer wavelength of light, not the others? I mean,
blue violet light was the intention of using, intention to be used. So
Okay,
that
so-
is why I choose 405
so
since
the
it
other,
was.
okay, so the other spectrum is not.
No, actually I used it because that was the first and foremost easily available wavelength with me.
Okay,
And
okay.
fortunately I could see the
So
results
you
at
can
405
compare
also.
with the other spectrum as
Yes,
well.
yes, sure, sir. The lower wavelengths.
Yes.
Yes, yes, sir. Yes.
Hello, congratulations regarding your work, very interesting. Can you comment about the bubbles formation on the microfluidic device, if you have it, and if yes, how are you avoiding?
Can you please be a little louder, please? I can't
Bubble
hear you.
formations on the bubble formation on the microfluidic.
Yeah, so I initially run the whole culture medium through it to ensure that there is no bubbles that have been accumulated. And yes, yes, yeah.
Thanks, Devashree.
Thank you so much. Thank you.